This video presentation is the Part 2 lecture on Newcastle disease, in continuation with Part 1. In this, we will discuss on the transmission, pathogenesis, diagnosis, prevention and control measures of Newcastle disease in detail. Before that, we will have a very brief overview about this infection. This disease is caused by the genus Aeulavirus, and species avian paramyxovirus type 1. This viral disease is highly contagious, and characterized by digestive, respiratory, or nervous signs, in domestic poultries like chickens, and in other bird species. Transmission. The nasal discharges, and the fecal excretions from the infected poultry, act as the principal source of this virus. This virus is having the portal of entry, through inhalation, and by ingestion route of virus entry. The incubation is on an average of 2 to 15 days from the entry of virus. Pathogenesis. Following entry of virus through inhalation, and ingestion, the virus does its initial replication in the epithelium of the respiratory tract and alimentary tract and at the regional lymph nodes, followed by viremia. And their next replication is at the brain, proventriculus, lung, spleen, and lymphoid organs. Following secondary replication, the virus gets distributed through the secondary viremia, with subsequent viral shedding through nasal discharges and fecal excretions. This virus survives in discharges and feces for weeks. 4 different forms of Newcastle disease are observed, based on the severity and the predilection site of the infection. Number 1. Viscerotropic velogenic form, also called as Doyle's form, where severe hemorrhagic lesions in gastrointestinal tract are observed. This form causes high mortality in flocks. This form also called as Asiatic or exotic Newcastle disease. Number 2. Neurotropic velogenic form, also called as Beech's form, where severe respiratory and neurological disease are observed. This form also causes high mortality in flocks. The velogenic Newcastle disease can either be viscerotropic or neurotropic, depending on its predilection site. Number 3. Mesogenic form, also called as Bodette's form, where moderate respiratory and neurological disease are observed. This form causes low mortality in flocks. And last, ledogenic form, also called as Hitchner's form, where mild respiratory tract infections are observed. Clinical manifestations. Also based on the severity and the predilection site of infection. Bird may be with respiratory signs, digestive signs like diarrhea, neurological signs, and other signs, like drop and egg productions are observed. Signs and symptoms are often misdiagnosed for other poultry diseases. Postmortem findings and gross lesions. Only velogenic strains produce gross lesions. And these lesions are not pathognomonic. Findings like swelling of periorbital area or entire head, edema of interstitial or paratracheal tissue of neck, petite cal hemorrhage on the mucosa of proventriculus, ulceration or necrosis of respiratory or digestive lymphoid tissue, and ulceration or necrosis of Peyer's patches are observed. The picture showing the pinpoint or petite cal hemorrhage in the proventriculus. Diagnosis can be done in field level or in the laboratories. Field diagnosis. Based on the history. Clinical symptoms. As we already discussed, signs and symptoms are often misdiagnosed for other poultry diseases. And postmortem findings. As we discussed earlier, only velogenic strains produce gross lesions. And these lesions are not pathognomonic. Next. Laboratory diagnosis. For lab diagnosis, oropharyngeal or tracheal swab, cloacal swab, serum are collected from live animal. Serum is collected to assess the antibody titer following vaccination, not used for diagnosis of Newcastle disease infection. In dead animal, spleen, lung, kidney, intestine, heart, brain, and liver are collected. This virus can be cultivated or isolated in lab by 2 ways. Number 1. Embryonated egg inoculation. 9 to 11 days old specific pathogen-free embryonated chicken eggs are used. For this allantoic route of inoculation method is done. Following inoculation and incubation, death of embryo is generally observed in 24 hours. On embryo observation, hemorrhages throughout the body particularly in occipital region are observed. Confirmation for the presence of Newcastle disease virus can be done by hemagglutination test, hemagglutination inhibition test, and by Revesse transcriptase PCR. The second method of cultivation of this virus is cell culture system. Primary cell cultures of avian origin are the most suitable for cultivation. Here, chicken embryo fibroblast cells, chicken embryo liver cells, chicken embryo kidney cells can be used. Next, Once the Newcastle disease virus is infected over the cells, the virus enters the host by fusing the viral envelope with the cell membrane. During fusion, the viral proteins like hemagglutinin, neuraminidase, and fusion proteins may get integrated to the cell membrane. So, this integrated fusion protein over the infected cell will make the infected cell to fuse with the nearby neighboring cells around, which is required for cell-to-cell spreading of virus. So after fusion with the neighboring cells, this appears like an irregular-shaped, multinucleated giant cell. This type of cytopathogenic characteristic is termed as syncytium. Here, the left picture showing the normal cells before Newcastle disease virus infection. The right picture showing the syncytia, which is generally observed 24 to 48 hours after virus infection to the cells. An irregular-shaped, multinucleated giant cell. This is a cytopathogenic characteristic of a paramyxovirus over the cells. These are some of the laboratory tests can be done for diagnosis of this virus. For antigen detection, hemagglutination test. Hemagglutination inhibition test using Newcastle disease virus-specific antiserum. Reverse transcriptase PCR and DNA sequencing can be done. For identification of this virus, the illustrated and prescribed methodology is followed. First, the process samples has to be isolated for the virus in the specific pathogen-free embryonated chicken egg. Following isolation through 2 to 3 blind passages, the allantoic fluids are tested for the virus by hemagglutination, hemagglutination inhibition using Newcastle disease virus-specific antiserum, and further confirmation by reverse transcriptase PCR and DNA sequencing. For confirmation, that is for viral RNA detection, reverse transcriptase PCR is done. Since it is RNA virus, first the RNA has to be reverse transcribed to complementary DNA using the reverse transcriptase enzyme. Later, this complementary DNA will be used to conduct the PCR. Prevention and control. 2 ways are followed. One is by medical prophylactic method, and the other is by sanitary prophylactic method. First we will discuss on medical prophylaxis. By vaccination. By using live attenuated vaccines. These vaccines are cultivated and attenuated in the specific pathogen-free embryonated eggs, and available in a freeze-dried forms. F strain or B1 strain or Lesotho strain or R2B strain are the strains used in the NDV vaccines in India and worldwide. Vaccination schedule. The primary vaccination is done at first week, using the lendogenic strains like F strain or B1 strain or Lesotho strain. F strain or B1 strain are the best choice for priming. Following primary vaccination, next booster is done at 3rd to 4th week age, using Lesotho strain. Lesotho strain are best to be used as a booster or revaccination, after the use of the F strain or B1 strain. Next booster is done at 8th week age using the R2B Muftesh or mesogenic strain. R2B mesogenic strain is used as a booster, after the use of lendogenic strains. And then the subsequent boosters and revaccination is carried out, with the Lesotho strain, as per the periodical antibody titer assessment, following each vaccination. For priming and booster doses, using the lendogenic strains, like F strain or B1 strain or Lesotho strain, is done through the intranasal or intraocular route, or by drinking water, and also by course spray method. For booster dose using the R2B Muftesh or mesogenic strain, is done through the intramuscular or subcutaneous route, or wing-web intradermal injection. In India, these are the types of Newcastle disease vaccines are available. Live attenuated lendogenic B1 strain, F strain, Lesotho strain, thymostable Lesotho strain vaccines. Live attenuated mesogenic R2B Muftesh or strain vaccine. And inactivated Lesotho strain vaccine. Thermostable vaccines are easily preserved and transported, because relatively they are resistant to inactivation on exposure to elevated temperatures. The first 5 are cultivated and attenuated in the specific pathogen-free embryonated eggs. And the last one is cultivated and virus inactivated using formaldehyde or beta-propiolactone. In breeding hens, inactivated multi-antigen vaccines are used against infectious bursal disease, Newcastle disease, infectious bronchitis, and reovirus. This inactivated vaccine will provide only humoral immunity, that is, antibody-mediated immunity, that too in a high level. So this provides paternal immunity, that is maternal-derived antibody, for the early protection of their progeny. There are also recombinant vaccines available for Newcastle disease, in which fowlpox virus, or vaccinia virus, or pigeonpox virus, or turkey herpes viral genome is ligated with the hemagglutinin, neuraminidase gene and the fusion gene of ND virus, and expressed. To assess the antibody titer following vaccination, antibody titration in the serum can be done by hemagglutination inhibition test using specific Newcastle disease virus, and ELISA. Here, the antibody titers are correlated, at the flock level, rather than at the level of individual birds. The hemagglutination inhibition antibody titer, equal to 1:32 or more than 1:32, are considered to be protective titer for Newcastle disease. If the antibody titer is less than 1:32, then they are considered to be unprotected. So, in that case the booster vaccination is recommended. Next. By sanitary prophylaxis. In this, following measures is to be established, to prevent all types of viral and communicable diseases in the poultry farming. Like, bird-proofing houses, proper feed and water supply, proper carcass disposal, pest control like, insects and mice control in flocks, avoidance of contact with birds of unknown health status, that is from newly acquired domesticated poultry, pet birds and wild or feral birds. Control of human traffic, like employees should not have contact with outside birds. Control of vehicle traffic. Strict disinfection of equipment. All in all out breeding, that is one age group per farm, is recommended. And disinfection between groups. During outbreak situations, effective quarantines and movement controls should be followed. Destruction of all infected and exposed birds, with a gap of 21 days before restocking. And thorough cleaning and disinfection of the premises. With this we are coming to the end of Newcastle disease. In next video presentation we will discuss on the rinderpest infection. Hope the lecture is informative and useful. Thank you.